Okay, I think we can get started since it's 1:05. Um, oops, there we go. Everyone can see my screen okay?
Yes, Margaret. And
Lovely.
just so you know, the other Margaret listed, I guess, is Colton. Somehow he has your name.
Oh, okay, cool. That's great. Awesome.
But
So
he
yeah,
has you in the chat.
wonderful. Thanks everyone for joining today's workshop, part two of the spatial transcriptomics workshop. My name's Margaret Ho. I'm a functional genomics specialist at BCBB. And I guess also on this call is Colton McNinch, my colleague. He might be posting as Margaret also. Um, He is a transcriptomics specialist at BCBB. Both of us have been seeing more NIH and NIAID researchers interested in spatial methods and actually generating data. So hopefully between the two of us, we'll be able to cover some of your questions. So please feel free to drop that in the chat or speak up. Um, yeah, so today's is a hands-on tutorial with Art and Seurat to analyze a example spatial Visium data set. You should have gotten an email from me earlier in the week with a link to the GitHub page. I also put it in the chat. Um, that has all the slides from part one and today's slides as well as our code that we're going to be working with later as well. Um, just to intro our branch, we're part of BCB branch at NIAID, and Mariam is one of our team leads. We offer scientific services and resources for NIAID researchers, and both Colton and I are part of the science support section. Um, it's organized into different teams based on our expertise, which is listed here. So if you're more interested in reaching out to us, please feel free to email us or check out our website. Okay, so today's learning objectives are to hopefully by the end of today, you'll understand some of the techniques that we use for processing and analyzing Visium spatial transcriptomics data with R and Seurat. You'll be able to understand the hierarchical structure that is used within Seurat objects to actually store our gene expression count data and the spatial data set. We will have a bit of a demo in the code for actually showing what count data looks like and how that looks like on a histogram. So you can see how it looks like versus other distributions like Poisson and negative binomial. You'll learn to filter your data in your Seurat object and normalize the count data using SE transform. And then we're going to go through a few steps of unsupervised analysis. So we'll be doing dimension reduction, clustering, visualizing those clusters with UMAP, and then doing a bit of differential gene expression analysis with DE markers. So we'll also be showing you different commands to actually like allow you to plot your data, plot your count data, your different genes that are in your data set, then different clusters, and we have different commands for doing that here. And then we'll do two types of downstream analyses today as well. That'll be identifying spatially variable genes using a metric called Moran's I. And then we're going to also be able to annotate our spatial data set using a matching single cell RNA-seq reference data set using another R package called RCTD. So what I have prepared for today is just a couple slides that introduce some of the method again and the different commands we're going to use so that you'll hopefully be able to spot them in the actual code. And then we'll actually get into running RStudio. So today's data that we're working with is 10x Visium data. So you may remember if you were able to attend part one, it's a spot array based spatial barcoding method. So each slide has four capture areas for placing the tissue slice on. Each of these capture areas has roughly 5,000 barcoded spots. And each spot has a oligo that contains a unique spatial barcode, shown here in green, a UMI, and is able to capture the transcript with a poly A tail. Um, the sequencing library that ends up being built from all of this is actually sequenced with paired end reads. So there's one read coming from one end that will capture that spatial barcode, and the other opposite read will capture a portion of the inserted transcript. And these spot arrays are around 55 microns in diameter and around 100 microns apart. So we expect each of these spots with a mammalian tissue to capture a mixture of between 1 to 10 cells. And 10x Genomics has recommended sequencing these in all these experiments to at least a depth of around 15,000 reads per spot. And so 10x Visium is able to do this on a whole transcriptome basis, which is great. So you see many, many genes, but the resolution's not that high, as you can see. And this is how this is how the Visium data set actually looks like with the picture of the tissue. So the data set we'll be working with today is a mouse brain data set. I'll have some more details about that in the next slide. But just to give you an idea of like how the actual workflow works, it's going to be fresh frozen slice that gets put onto the capture area on the tissue or on the slide. And there's immunofluorescent staining or imaging that's performed to get a sense of the histology and also to orient the tissue properly on the slide. Then there's permeabilization of the cells to allow those spot array oligos to penetrate and capture the poly A transcripts. And then there's reverse transcriptase reaction to make cDNA, and then amplification of that cDNA, and to actually generate the sequencing library. And we are working with Visium data. This is the original Visium methodology because the data set's actually light enough to be run on local RStudio on your laptop. So this is the data set we'll be analyzing. It's a fresh frozen mouse brain slice, which is sliced sagittally. This is what the tissue looks like on the slide. And you can see, hopefully, I'll actually pull it up in a bigger resolution later. You'll see the surrounding dots on the edge of the capture area that's used to calibrate and position the image. This allows everything to be oriented precisely so that we can actually get the precise XY locations of every barcoded spot. And 10x Genomics, when they generate this data and run it through Space Ranger, it will provide a web summary to actually show some of the details about the data set. So I'm going to try and pull that up here on the left. Hopefully you should see the web summary. So yeah, if you hover over this, is everyone able to see the web summary okay? You should be able to see the tissue oriented on the array, and then there are these red calibration spots that actually allow us to know exactly what the XY location will be of each spot corresponding to the image. So there's roughly 3,300 spots that are captured over this tissue. And then there's 87,000 reads per spot, which is actually well above that 15K minimum we talked about before. Roughly 4,500 genes in total detected per spot. And then the reads are 292 million reads over the whole experiment. And then there's data that shows the percentage of valid barcodes, valid UMIs, and then when they map the reads to the genome or to the transcriptome, it shows you some stats of what percentage are correctly mapped. So we're getting great high percentages of mapping to the mouse genome and to the mouse transcriptome here. So hopefully you can see that. So this is just more metadata that's provided by 10x Genomics about their data set. You can see that the tissue image is actually captured with a pretty simple color microscope. And then this sequencing is done with paired end reads. Okay, so after the image and sequencing data is collected, it gets pre-processed in a software pipeline called Space Ranger, which is provided by 10x Genomics. If you've worked with single-cell RNA-seq data, it's very similar to Cell Ranger. The purpose of Space Ranger is to detect the tissue, place the spots in the spatial context on the slide image, um, align the reads to the reference sequence into a transcriptome, and then generate feature spot count matrices. So that'll be the genes versus spot count matrix. And the main programs to be aware of here, because this is like a lot of different modules, is basically Space Ranger Count that actually generates the count matrix, and then Space Ranger AGGR, which aggregates data across all the slices, because there's multiple slices on the capture slide and outputs of files. So Space Ranger can also perform some preliminary clustering and gene expression analysis, but we're going to end up doing that ourselves in Seurat. On the right are the output files from Space Ranger AGGR. You can see the web summary over here that I just showed you. And then the other two things to be aware of are this folder called Filtered Feature Barcode Matrix that contains a list of the barcodes, a list of the features, the sparse matrix with the count data, and that all gets wrapped up into one file called filtered feature bc matrix.h5. That's actually what we're going to feed to Seurat for the analysis. And then the other thing to know is there's going to be this folder called spatial, and each of these would represent a single slice. And then it has images of the slice, as well as some information about scale factors that allow it to know how much to scale the image relative to the location of the spots. So yeah, we're going to load this data into Seurat. When you do that, you would provide the H5 file and then the name of the slice that you're working with. So this is today's workflow. We're going to be providing this filtered featured barcode matrix, which essentially is this, it's going to be spots versus genes count matrix. And then it also has, because this is spatial data, it's going to be the spots versus X, Y coordinates of the cells. And then we're going to perform several different pre-processing steps. We're going to normalize the count data in Seurat. Then we'll be performing dimension reduction with principal components analysis, PCA, cluster the data, and then visualize those clusters on a UMAP. Then we'll be doing some downstream analyses of the data, two different kinds. So we'll be doing analysis of spatially variable genes and identifying those using Moran's eye. And then we're also going to combine in a reference single-cell RNA-seq dataset to do cell type annotation with a program called RCTD. So I'm going to go through a few of the commands. This is just so that hopefully you'll be able to spot them in the R code. So we've got this specialized command to load the Visium data. It's called load 10x underscore spatial. We're going to provide that data directory that I showed you before, that Space Ranger AGGR would provide. You would then point it at the appropriate filtered featured barcode matrix H5 file. Then we tell it to look in the assay's name spatial, the folder name spatial, and we're telling it to look at slice 1. And actually, when we actually do this, we actually have a helper data set, so we don't actually do this, but just know that if you were loading in your own Visium data set, you would be actually providing those specific file names. This is for Visium data and Visium HD data. If you were loading like Merfish data or Xenium data, you would have other commands like load visgen and load Xenium for those. And then just down here, I'll show it to you when we actually run the code, but there's a great command called utils method class Seurat, which will actually show you all the different commands that you can run for Seurat objects. And that's kind of helpful to know what other things you can do within Seurat on your data once you've loaded it in. So these are other Seurat commands that you will end up seeing in the R code. We're going to essentially be doing pre-processing of the data. And this is a type of analysis that's called unsupervised analysis. It's very similar to what's being done in single-cell RNA-seq analysis as well. So we have count data that needs to be normalized. we have essentially excess variance in the data, and SE transform is the algorithm within Seurat that's available to us to normalize that data. Once we do that, we can then do dimension reduction, and we'll do that with PCA, and we're going to run it on the normalized count. So that's why here we're going to provide it the Seurat object, and then also tell it to use the assay that's called SAT, because we want it to do it on the normalization on the normalized counts. Then we'll be doing clustering, and to do that, we have to build this shared nearest neighbors graph. So we do that with find neighbors. We would then provide the Seurat object, and then there's parameters that tell us, oh, okay, run this on the PCA reduction that we did in the last step, and then we'll tell it to run on the first 30 dimensions. And then you'll actually run find clusters to identify the clusters. Those clusters can then be visualized on a graph using an UMAP embedding. And so you do that by running UMAP, and you tell it, okay, run the clusters using the PCA reduction. And then the last thing we'll do is actually find differentially expressed genes between different clusters. And that'll be done using Wilcoxon rank sum test and find markers. Great. So, okay, these are just a couple more commands that you'll see that are actually specifically to plot our data. These are two commands, spatial feature plot and spatial dim plot, that allow plotting of the data over a tissue histology image. Feature plot refers to feature is gene, so it's going to plot the actual gene expression over the tissue histology image. Spatial dim plot is referring to clusters or different categorical groupings. It'll allow us to plot the clusters on that histology image. If you don't want to have it on the tissue histology image, you can also use what's called image plotting. So there's image feature plot, image dim plot, same thing as before, except it's plotting on a cartoon essentially or a spatial map of the different spots or cells. And it's going to be allowing you to map or plot the features or the clusters again. Very similar. Okay, so those are some of the commands we're going to do. I'm also going to introduce some of the commands for the downstream analysis. So the first downstream analysis that we'll do is annotation with a reference single-cell RNA-seq data set using RCTD. And RCTD is a R package that's from Rafa Irizarry's lab. It stands for Robust Cell Type Decomposition. I mentioned before that each Visium spot is going to encapsulate multiple cells. So in order to actually determine the cell identities of the that make up each spot, we have to use appropriate reference single-cell RNA-seq data set. This will allow us to label each spot or cell in the spatial data set. And this whole process is called deconvolution. So RCTD will take this spatial data set, in our case, our Visium data set, and the single-cell RNA-seq reference. You build a reference-based probabilistic model to essentially assign a cell type to the spot using maximum likelihood. And we'll run it in doublet mode, which allows it to make its best guess of either one cell type that it thinks makes up the spot, or maybe a proportion of two cell types. Then we'll end up with each cell in the spatial data set as annotated with a likely proportion of those cell types. So to actually run RCTD, I wanted to break down the commands just because it can be a little hard to understand in the R code. But essentially, we need to take our single cell reference data set and create a reference object from that. And then we're going to take the Visium data set and make a query object from that, from the spatial data set. And we are actually going to calculate all these different components and then feed it to the reference, this function that is going to create the reference object. So we're going to give it a count matrix, and this count matrix is essentially genes on the rows and cells on the columns. That'll be fed here. And then we're going to give it a vector that is the cell type annotations for every cell. And then we're going to give it the total counts of transcripts observed for self. This is all from the single cell RNA-seq reference. Then for the spatial data, for the Visium data, we're going to do the same thing, but we're going to provide the spot coordinates as coords, the count matrix again, which is the genes versus barcodes and the counts, and then the number of total counts observed per barcoded spot. So we're going to essentially feed it each of these three things here to make the reference object and the query object. And then there's a command called create RCTD that essentially we feed the reference object into here and the query object, tell it to exclude cells from the single cell RNA-seq data set that are fewer than 10. And then we'll actually run RCTD on that. And that'll take a little while, but we'll run that. And I have some of the results saved as well in case it doesn't finish in time, but we'll run it in the doublet mode. Okay, so the last bit here is the last bit of downstream analysis is looking at spatial variability in gene expression, which can be measured using a metric called Moran's I, which measures spatial autocorrelation. Moran's I is going to range from 1 for positive spatial autocorrelation. So the idea is that for a particular gene, there might be cell neighbors that show similar gene expression for that gene, whereas -1 would show negative spatial autocorrelation, where neighbors are showing opposing gene expression, or you might have no spatial autocorrelation, or autocorrelation, which would be 0. And there's a command to run this called find spatially variable genes. It will specifically detect those genes whose variability and expression can be explained by spatial location, and it will tell it to use the selection method Moran's I for that. Okay, so those are our steps that we're going to go through, and hopefully highlighting some of the commands so that you'll be able to spot what we're doing at every step. So any questions? I can move on to RStudio in a minute. I'm just going to quickly check the chat. Okay. Let me just quickly turn off my notifications because I think here, I'll just close Teams. There we go. Great. Okay. So here's our workspace in RStudio. I'm going to clear the environment so that we're starting from a clean slate. So you should have been able to open the Quarto Markdown document in the GitHub and open this. This will show our R code. It also has a lot of information as well to guide us through. So first, what we're going to do is actually, you should have run some of this ahead of time in red, hopefully. If not, I provided the PDF for people to follow along. Let me pull that up actually as well. And you should have been able to essentially run this part in red, which would have essentially installed a lot of the packages ahead of time, like Seurat data, RCTD, other packages that help us visualize and plot data. So we're just going to load those libraries right now. We have Seurat data. These are libraries to help plot and analyze data, like tidyr, ggplot2, etc. We're also going to load RCTD. And you should have also downloaded ahead of time the single-cell RNA-seq data set that we're using, which is a mouse cortex reference, which is basically just a portion of the cells. It's going to represent a portion of the cells in the mouse brain that we're expecting to see. And there are also some libraries that are helpful to run these packages. To run these packages quickly. So there's some information here about actually understanding how the Seurat object is structured. I'm going to load in our data just quickly. So here, this section, if you're able to follow, we're loading the data using the STX brain package, and we're telling it to use anterior slice 1. Specifically, so it's loaded it in, and actually I told it to run brain at assays. That actually tells us what assays we have available. So if you open here, the R object, it's going to be huge hierarchical data structure. It has a lot of slots. These slots are going to get populated as we run our, as we pre-process the data. So currently it has, so if you look at the metadata for this, it's got the, uh, the transcript counts for every spot. They've also got the number of genes observed for every spot. So N count spatial is the number of transcripts, N feature spatial number of genes. And then currently right now the spots are showing, let me just check. Um, Currently we're showing that the identity of the slice is just slice 1 and interior 1. And currently there's no cell identities assigned to the spots. So it's just original ident and it just says 1, 1, 1, 1, 1. As we run our pre-processing data, these things will start filling out. We're going to get another assay that's going to get filled in here called SET. That'll be our normalized data. And then things are going to start populated here as well. So that we'll have, where is it? We'll have reductions, for example, or neighbors or graphs populated in. So this Seurat object is this hierarchical data structure. It has tons of layers and it gets, as we're running things, it's storing it in different slots in the object. And that's described here. There are different slots, assays, metadata, active assay, graphs, reductions, et cetera, that you can look at. And if you wanted to just look, like if you didn't want to open this object right here on the right, you could actually just type in here. Hopefully you can see. It's a little bit hard for me to see. Okay. Here you could actually like start looking in here. So if you go it will actually show you what's available. So these are all the different slots. So in this case, we had brain assays. And it shows me a little bit of information. We have one assay, which is the spatial assay. That's the count data. It has 31,000 genes that we're observing and roughly 2,696 cells. And it shows some of the names of the genes that we're looking at, and the layer it's looking at is called counts. Great, okay. Again, this one is showing here utils method class Seurat. If we run that, it shows this, which is just different commands that you could run on your Seurat object. So I think that's somewhat helpful. We will be running a lot of these, like find spatially variable features, normalize data, et cetera. It just shows you what is the tool set available for running commands on your Seurat object. Thanks, folks, for sharing the stuff in the chat to get people up to speed. Okay, so we loaded the data. We're not going to use this load 10x spatial. So yeah, if you just actually look at the number of rows in the brain and the number of columns, it actually shows you the number of genes that we observe and the number of spots, right? So that's just actually shown as like the dimensions of the Seurat object. This is a great thing to run in case you don't want to open this whole thing. The summarize is very similar, but it summarizes everything that's on the right here. If you didn't want to open your data object, if we type in glimpse brain, it essentially shows here that brain is a Seurat object, and it has all these different things summarized. So that's kind of helpful if you don't want to read through everything, because sometimes it gets cut off at the bottom. We talked a little bit about the metadata. I think, did I already show this? Yeah, if you looked at metadata, you're going to actually see what the structure of the spatial count is. And so it's just showing the same thing that we saw on the right here. Yeah, if we looked at row names, I think that would be the barcodes. Yeah, so these are all the individual barcodes that are labels for the individual spots, kind of equivalent to the cells in a single cell RNA-seq data set. Okay, so we're actually going to do some plotting here. So we can actually look at the distribution of counts that is observed across all of the cells, and we can do that with violin plot. Sorry, some of this is already run prior to, so you'll actually see some of the results ahead of time. But if we run violin plot and actually tell it to look at the brain Seurat object, we tell it to look at specifically the number of transcripts. It will actually show us that typically we're seeing 30,000 transcripts per cell. And you can also look at how that looks like with spatial feature plot here. So if we show that individually, this is actually showing the number of transcripts observed in each spot across the entire brain. And it's since this is spatial feature plot, this is plotted over the tissue histology image. So this is just code that kind of wraps it all together in one plot. So if you run this, it should give me the plot. Yeah, it will show both plots together. Great. So just to orient you a little bit on the mouse brain that we're looking at, we don't actually need to know all these different details, but over here we're looking at a sagittal view, and the main thing to look at is that we're going to be working with looking at some of the interesting neurons in the different layers of the cerebral cortex. That's going to end up showing up here. And then here is we know this is going to be a lot of white matter within the brain. This is the olfactory bulb, etc. So I just want to demonstrate what we mean, because I talked about in part one about what we mean by overdispersion of count data. So this is the counts of all the transcripts that we're observing. So I'm going to actually make a variable called endCountSpatial and pull in the count data into that. And so you actually see what I'm saving here. So this is the count data from the brain object. And you can see all these numbers. We're essentially going to plot a histogram of all of that. So that's the number of counts observed across all of the cells. And then we're saving to a data frame and we're just plotting as a histogram. So that's what's shown here. And we can see it's unimodal. If we wanted to model this with a Poisson distribution, essentially a normal distribution, we would essentially so what we're trying to do here is actually create a Poisson distribution with the same mean as our observed count distribution. So here I'm calculating the mean of our count data, saving it as lambda, and using lambda to plot an equivalent Poisson distribution of our with the same mean as our count data. And so what you can see here is, so the Poisson is shown in red, and then our data, our observed data is shown here in blue. You can see that the Poisson is super, super skinny, and our observed spatial data is super, super wide, telling us that we have a ton of variation in in the observation of the count data. And so you can get a sense here that the Poisson distribution is not appropriate for modeling our count data. It's way, way higher variability and way higher, way over dispersed. So we're actually so what people end up doing to do normalization is actually model the data with negative binomial. And so we're going to show something like that here. So we're calculating not just the mean of our count data, but we're also calculating the variance. And then we're going to feed that to a negative binomial distribution. First, we have to calculate the dispersion parameter for the negative binomial distribution from these two things, the mean and the variance. And then we're going to feed that to a negative binomial distribution. Basically what they show with here is, so our data here is in blue, and the modeled data with a negative binomial distribution is shown in red. And so you can see it's a lot closer, right? Like we're not having this skinny, skinny peak trying to model our varied count data. They're actually pretty close to one another. And so that shows that negative binomial distribution is going to be a lot better for modeling our data. And that's actually how partly how the regression works when we do the normalization. So if we look at the standard deviation of the count data and our negative binomial data, they're very, very similar. The standard distribution is very, very close. So basically the point is that when we model our count data with a negative binomial distribution, the fitted model only slightly underestimates the variance. And so we can use normalization with negative binomial. So we'll be doing that with SE transform, which is using a regularized negative binomial regression to get rid of that variance. So the first thing we'll do before even that is checking for mitochondrial contamination. And so if you've done some single cell analysis, you know that this is something you have to do. Essentially, when you have a lot of mitochondrial genes showing up in your data, it's often an indication that there was some kind of cell death or stress that the cells are undergoing. So we want to actually remove cells in which mitochondrial genes are a huge component. So we can actually look at the percentage of cells in which so we can actually plot the amount of mitochondrial contamination that we have. So we can actually make a variable in the metadata called %mt, um, and set it to and grab the percent of genes that start with MT. That's how we know they're mitochondrial genes, and then do a violin plot of that. So when we do that, we see that most of the genes have about 15% of mitochondrial genes. Um, but there's going to be a small percentage of cells in which there are in which there's way more mitochondrial contamination. So we're actually going to eliminate some of that. And I think there's a question in the chat, removing mitochondrial genes, wouldn't that be an issue in disease states? Potentially. I mean, this is a choice that you have to make, whether or not you do it. But I think just for the purposes of this, in general, it is good to look at the amount of mitochondrial gene contamination you have, because that can help you decide whether it's something you want to do. If it ends up removing a ton of cells, maybe you think that's actually related to your disease and you might not want to do that. Um, okay, so I actually like to see how the mitochondrial contamination is. I prefer to actually see it on a histogram. So here is actually showing a histogram of that, and then you can see that there's this long tail of some cells, a small percentage of cells that have more mitochondrial gene component than expected. So we're just going to cut off around like 28%. So this is a command to do that. We're going to subset brain, the object brain, using that percent mitochondria as which I believe you should actually see now here in meta. Yeah, so I don't know if you can see this, but at metadata, we've created this %mt now that tells you the percentage per cell. So we're actually going to be filtering on that to remove um, uh, any cells that are higher than 28%. This is arbitrarily chosen. Yeah. Because we just want to filter out maybe some cells that we think could be slightly disrupted without actually affecting our analysis too much, but we're just trying to be on the safe side. So we can do that. We're going to run the subsets, and then we're going to create a new object called brain empty filt. So now everything's going to be done on brain empty filt. So you can see that this is the Seurat object for that. Then we can actually run our SE transform. And here, actually, I think what I did was I was just plotting the data, and it looks very, very similar even after filtering the data, because this is run on brain empty filt. So we're going to run SE transform on brain empty filt, and I'm just going to start running that because it does take some time. We're telling it to run it on assay spatial, which is our count data. I'm telling it to be as verbose as possible to tell me lots of things. You can do the subsetting to subset your data to get rid of the mitochondrial data ahead of time, or you can actually also play around with the regression. You could you could try and regress out some of that percent mitochondrial data if you wanted to. So that's one option you can do if there was something you wanted to regress out, if you thought that that was a huge source of variability in your data and you didn't want to actually include that. So it's run already. It's done the negative binomial regression. There are a bunch of parameters. What you'll see is that if we actually look at the assays now, we look at brain empty filt, there's now, in addition to this spatial assay that we had originally, we have the SCT, um, the SCT normalized counts. So yep, that's shown here. You can also see that if you started playing around in here to look, where's the console? We're going to look at empty filt. And if you looked at assays, now you can see that there's not just spatial, there's also this SET assay. So it's actually slightly different. So a bunch of genes were removed actually, because I guess they're not very variable. And then so yeah, we have a fewer number of genes for that, but essentially is able to also identify highly variable genes from this analysis. Right, so I'm going to show you some of the plot data. So we can use spatial feature plot, and here we're plotting genes HPCA and TTR, which are different neuronal specific calcium binding proteins or a different transporter for retinol. Here we actually can see the spatial expression of genes on the tissue. And you can actually play around with these parameters of how you want them to look like. So you could play around the plotting parameters. You could change the point size factors or the amount of transparency in the plot. So we can play around with that. So here we're plotting again to show more transparency to actually see more of the tissue histology image. We also talked about image feature plot or image plots. These are going to show this not on the tissue histology image, but on a cartoon. Here I'm plotting TTR again, just so you can see it, how that looks. But then you can also, in this case, I'm plotting GFAP because it's an astroglial marker. We know that it's very strong in these white matter areas, so it's kind of neat to see that show up. So we can see the higher expression areas of GFAP in those spots. So then we can perform, after we're doing normalization, we can perform other processing steps to do unsupervised clustering analysis. So we'll have to do some dimension reduction with the PCA to do that, clustering and visualizing those clusters with UMAP. This actually takes a little bit of time, so I'm just going to run this. Actually, this one's not too bad. So what you can see here is that it ran a PCA, and here it's listing different principal components. So PC1, it's showing all these genes would be kind of similarly correlated in one direction, in the positive direction. This is in the negative direction. So it's going to run principal components analysis to essentially reduce the number of dimensions that we have in the data set. Then to do clustering, we can run it directly on the PCA data. So we're telling it to first make the shared neighbors map with the PCA reduction and run it on the first 30 dimensions. And then we ran find clusters on that. So we are able to identify the different clusters. The default resolution on this is 0.8. And something I also wanted to mention just for folks who are new with R is that you can actually, if you want to know how to run some of these functions and what are all the different parameters involved with them, you can actually, for example, grab one of these and actually run question mark in front of them. And if you do that, it will actually show you information about how to actually run them in additional parameters. 0.8 is in reference to the resolution of the clustering. So you can actually run this with multiple different types of clustering resolutions to kind of allow you to do, and I believe this is this is in the clustering. So yeah, if you do find clusters, you can actually kind of modify the way that it's finding the clusters. How much 0.8 is specifically like the modularity that's used to define the clusters. So you can, that's just one way that you can, as you're running these, you can actually try different parameters. Okay, so we ran all of this. It also ran the UMAP as well. So what we're going to see afterwards is when we actually run this, reductions, rain empty filt, if you just want to see what's here, it shows us now that it has a PCA reduction and a UMAP reduction available. And we're going to be plotting those in just a sec. But you can see that also here in this rot object. If you check in the metadata now, we now have information about the clusters that it's actually assigned. So every spot is going to get cluster assignment. Then we also see the graph being added, and it's actually not showing me the reductions because it got cut off, but we can see it just by checking what the reductions are on the object here. So we've done our PCA and we've done our UMAP. We can actually, this here, I'm just looking at some of the information from the PCA. It ran it with 50 different principal components. It ran it on the SCT normalized data. Here, if you just look at the brain-empty filled clusters, you can see every single cluster that each spot was assigned to. So this is the barcode for every spot. You can say that this spot was cluster 6, etc. So if we run summary on that, we can actually look and see we have 14 clusters. And then this is the number of cells that actually ended up in each cluster. And so we can do plotting now. Everyone always wants to see plots. So if we do dim plot, we can specify it to look at the PCA. We can actually look at the PCA plot, and this is for the first two principal components dimensions. And then if we run spatial dim plot with brain empty filled and tell it, and actually we just run it the way that it knows that it's in different clusters now. So it's actually going to show us the spatial location of every cluster. So that's kind of neat. You can see them side by side as to which cluster goes to which area on the actual tissue. And then because PCA is not the most interpretable thing, we did UMAP embedding. So we can actually run that. That will show us the actual embedding of the clusters on the two-dimensional space. Yes, you can change the colors. I think I'm just leaving it at defaults to keep the code as simple as possible. Yeah, if you're interested in all of this, these plots are essentially ggplot plots. So if you want to learn how to change colors and things like that, you just have to add in additional ggplot parameters for that. I can provide information on that later. But yeah, so this is just keeping it to the default. So we have all this, we've run all this processing now on our data. We want to, like it's quite a bit of work. It's very helpful to save your data as an RDS object or our data set object to load again later. So that's done here if you want to do that. What we're going to do now is just show you a bit more about plotting. You can actually identify the different clusters with spatial dim plot. Here, I'm adding in a little bit more information. So yeah, you could modify the colors in here. For example, I'm just keeping it to the default. Here, I'm adding additional grid lines. So I think that's kind of helpful if you wanted to do cropping of the image, for example, which we're not going to do here. But so here's actually showing the X and Y coordinates of every spot. If you wanted to say only grab, the olfactory lobe or something, you could actually use the crop command to do that. So that's this command. This is part of Seurat. So yeah, this is the command that you could use. And you could provide X and Y coordinate ranges to crop on. Okay, so that's spatial dim plot. So it's showing the clusters on the tissue histology image. Then there's image dim plot, but then it shows it as a cartoon. So that's kind of cool to see. And then other functions, if you want to play further, there are ways to do this interactively. So if I'm going to uncomment this stuff, so actually it's going to show up here. It's same spatial dim plot. The only change that I made here is that I made interactive equals true. So now if you hovered over one of these spots, it would tell you what cluster it belongs to. So this is part of group 0, group 2, group 6, etc. That's just fun to play with. Um, you can click done to properly end that. And then spatial feature plot also has an interactive option as well. Interactive equals true. Here you can actually play around with aspects of the visualization, like depending on how strong you want the colors to be, the intensity, and the different point size, the different transparencies available. How do you keep the there's a way to look at the Seurat version. I don't have it here, but you can actually report the Seurat version. Oops. Can't remember if it's this command. Let me get back to that. But there's a way you can just report the amount of the version that you're using, and that's something you should do every time that you run Seurat. Let me write that down. Just to add, I'll address at the end of the tutorial. Go back to where I was. Okay, so spatial dim plot, image dim plot, the interactive features. The last interactive one that I think is helpful to look at is called linked dim plot. This will show both the spatial dim plot showing the different clusters and the UMAP. So if you wanted to click on the UMAP, you could actually highlight all the different clusters that you're seeing, what that actually corresponds to. Similarly, I believe you can also, yeah, you can click on here and see what cluster that corresponds to as well. Okay. All right, so something that was interesting from running this analysis is that we actually have different layers in the cortex that were found even just from the unsupervised analysis. So if we wanted to actually look at, try to do differential expression analysis between these different clusters, we could do that. So if we wanted to find differential expression genes, genes are differentially expressed between, say, cluster 2 and cluster 6, or cluster 2 and cluster 4, you can run that. That's going to be shown in this section here. So here I'm running spatial dim plot. I actually highlight each individual slice, each individual cluster, just to see how they're spatially laid out. Oh, and this is just showing the violin plot that we did earlier. Because we ran a SET transnormalization, it's going to actually show the UMI counts across all of the different clusters. That's kind of just helpful to see. So here I'm talked about doing the differential gene expression analysis. We load a library called Presto, just makes things a lot faster. This takes a little while, so I'm going to run it. Here I'm using the command called find markers, which I mentioned to you before. I provide the Seurat object. I provide the identity of the cluster that I want to operate on. So the first cluster and then the second cluster. So I've picked cluster 2 and cluster 5. It's going to perform differential gene expression analysis on these two clusters and try to find genes that are differentially expressed between these two, either more expressed in 2 or more expressed in 5. And then here I'm plotting the first 3 that I get from here. So I've saved it under DE markers to 5. We can actually look at that more detailed. So here I'm just looking at the first couple genes. If you just wanted to, if you're confused by that, here, I'll just do it here. It gives you a list of genes. This is why I used head. It's because there's a lot of them. But the way that it's formatted is that it's going to show you a unadjusted p-value, then a log-fold change of the average expression between the groups. So it's like -5-fold more expressed in one than the other. It's expressed in all of the cells of cluster group 5, and it's only expressed in 30% of cluster group 2. And here's the adjusted p-value using Bonferroni. So what's helpful about that is these are differentially expressed genes now that are between these two different clusters. You can also just start looking at them and see that, yes, cluster 5, I believe, was the olfactory bulb. Cluster 2 was one of the layers. in the cortex. So you can see that these genes are either expressed in one or the other. You can also find markers just on a given cluster by itself. So here we go to find markers. You'll see that one of the options is you can put cells 2 as null. So vector of cell names between group 1 and group 2. I believe in here. Right, so you can provide identity class 1. You could also put null. And if that's the case, if I put one cluster here and nothing here, it would just try and find differentially expressed genes in that cluster 1 versus all of the clusters. So you can run it that way as well, not between 2 clusters, but between one cluster and all the clusters as well. Great. Thanks for answering the question about versioning. Yeah, it's actually a more complicated discussion, but you can at least report and record down what version you're using. And if you wanted to actually take better cares to all the versions for your R packages, you can use something like R environment to keep that consistent. So we've done mostly our pre-processing. We've done our normalization, dimension reduction, embedding. We found the clusters, we've mapped the clusters, we've done differential gene expression between those different clusters. The other thing that we can do is find spatially variable genes. This is going to be using the command find spatially variable features. And we can specify specifically which method to use. We're going to use the method called Moran's I. So here I'm providing the object, telling it to run it on the normalized data, telling it specifically actually run it on the 1,000 most variable genes, so features. So we used a command called variable features to figure that out, provide that to the find spatially variable genes, find spatially variable features command. So that's going to run for a little bit. And then it's going to end up adding a column in the meta features that shows the Moran's I. So, okay, it ran. And so here, what is this? Oh, yeah, this is the normalized data here. So if you can see here in brain empty filt, there is now a meta features data frame, and it has information on the observed Moran's I, the p-value of what it's observed, and the rank of that gene. So here it is here. Within the normalized counts, it's got this this layer called meta features. It's a table, and it actually most of it is blank because actually a lot of the genes are not spatially variable. So I'm actually going to here I run it through NA exclude and save it to a data frame, and then we show that table again. So everything that it was blank because it's not a spatially variable gene that got removed. Now we're only looking at spatially variable genes that I was able to identify. Uh, so I believe it runs spatially variable gene analysis on all of the genes individually, and then it shows a p-value, and then it essentially is looking for genes above a certain cutoff. So here it's showing the observed Moran's I. So I mentioned before that it can range from 1 to -1. So because it's positive here, we can see that this is one that it was observed to be spatially autocorrelated. And then it also shows a p-value for that, and it also shows the rank. So this is the #1 ranked spatially variable gene based on p-value, and then it goes all the way down to, many, many genes. So you can use spatial feature plot again to actually plot that now. Here I'm grabbing the top, what am I doing? I'm grabbing the top clusters. Oh yeah, there's some funny way that this table is laid out that made me do this. So yeah, it takes the top 6 most actually the top 5, because the rank is less than 6. It plots the expression of that gene across the sample. So you can see that some of them are interesting, some of them maybe not as not as spatially restricted. But I think I talked in part one about some of the limitations of this method, but it is one way to find genes that have interesting spatial patterns. Okay, so that's one type of analysis. The last thing that we're going to do is make use of our single cell RNA-seq data set. So we have reference single cell RNA-seq data set for the cortex. We have it for a lot of different regions, but one of the great things about this particular slice is that we can see these different layers. So one of the things we can ask is what can we use the single cell RNA-seq data set to better label them and actually delineate the different layers. So we can do this on our whole data set. It would actually take a really long time. What we're going to do first is subset the brain empty field data set to just the clusters that are actually in the cortex layers. So out of all of those unsupervised clusters, number 2, 3, 4, and 6 were actually, or in part of 1, actually covers part of the cortex. So we're going to just not, for the purposes of this analysis, we're actually not going to analyze or run the RCTD on anything that's not in those areas. So we're going to subset brain empty field with those cluster names 1, 2, 3, 4, and 6. And that's going to be called cortex here. And then here it is just plotting what I've grabbed essentially. So we'll run the subset. And then we have to perform renormalization of everything. And this actually yields like clusters that are more defined. It's actually going to take a while to run, but yeah. So it did reclustering. This is a U map that shows everything. If you compare what we had before, we've got 1, 2, 3, 4, 5 here. We actually get more defined clusters. So now number 1 gets divided up into other clusters. It's better delineating the area where there would be white matter with the first interneuron layer. So we're actually seeing that now we've properly separated them out. But so that was just illustrating what you could do with additional reclustering. But what we can do with annotation or the single cell RNA-seq data set is actually do our deconvolution. So I'm going to load our single cell RNA-seq data set, and then you should have downloaded this ahead of time, Allen Cortex RDS. We then get another, I know there's a lot of stuff here, but I'm going to close some of this. We now have a Seurat object that's representing our single cell RNA-seq data set. It's showing the RNA and the counts data. We're actually going to grab all of that. So if we grab from reference within class the assay RNA, we're going to grab the counts. So you can actually see what that looks like once I run this. So ref counts is one of them. It's going to look like that sparse matrix of counts. We actually grab the cluster, the subclass. So within here, within ref, there's a lot of metadata. We can see the subclass. So each of these cells is assigned an identity, and it should be under subclass. VIP, LAMP5, etc. It also has information about like if it's GABAergic, etc. But essentially, this is a proper annotation that's within our single cell RNA-seq data set. We'd actually like to be able to transfer that onto our spatial data set. So yeah, we basically calculated the 3 things we need for our reference. We've got the counts data, the cell type data, and then we're also calculating the number of UMIs, which is just the number of transcripts observed in each cell. And these are cell identity numbers. Similarly, we're also going to take our spatial data set, and this is actually an additional step. I don't always do this personally, but for the purposes, I think, of our class, it is helpful because it does allow us to what's called sketching the spatial data set. So what it does is it basically downsamples some of that data. So we're only going to do the RCTD on a small portion of the spatial data set. And then we're going to essentially transfer those labels back to the large Visium data set. So this is calculating that here. So that is, I believe, it's been sketched. It's going to get an assay named sketch within it. So what I did here is I set default cortex as sketch. So now we're going to be operating on the default with sketch as the default assay. And then I'm going to essentially renormalize everything with assay equals sketch. So that sketch layer within cortex is going to have all this, all the pre-processing done on it. And so should be a sketch. Yep. So if you look at cortex assays, we've got a spatial, the original count data, the normalized data, and then we have a sketch data set as well. So that's all run. And then, yeah, so we calculated the reference stuff. We're providing those three things, the count data, the cell annotations, and the NUMI to create the reference object. And then we're going to do the same for the query object. So we're looking at cortex, we're looking at specifically the sketch assay, and then we're going to get the count data from that. So we're going to run this. So this is the count data. It looks really weird because it's all in sparse matrix format. This is the spot names and all the barcodes. And then we're going to give it the coordinates of the spots. And we use something called get tissue coordinates for that, just to get the coordinates. And that'll show X and Y, essentially, coordinates for every spot. So we give those three things to spatial RNA. That'll create the query object. I believe I ran that already, but we'll run it anyway. And then this is the part that takes a really long time. I don't know if it's actually going to run. I might actually skip this just because it takes several minutes. But essentially what we're going to do is you create the RCTD object, we run query, we run reference, we give it the query, we give it the reference. It's going to create this RCTD object, and then we're going to run RCTD, and then it's going to take a really long time. But I'll click it just so you can see what it looks like. Here it's telling me these are all the different cells that are in the single cell RNA-seq dataset. This was under subclass before. So it has a bunch of astrocytes, oligodendrocytes, parvalbumin neurons, different interneurons, et cetera, et cetera. This is going to take a long, long, long time, so I'm going to skip it still. But yeah, we'll start workers, and it will essentially be running RCTD for many, many minutes. What I end up doing with RCTD after it's run is that I look at there's going to be a metadata column for RCTD object, and then it's going to show some of the results from that. And I'm going to save it as metadata to Cortex. I'm going to skip this because it's going to take forever. I saved everything, I believe, at the bottom. Yeah. So let me uncomment that. So I'm going to read in the RCTD after it's been run. Just pretend that happened very fast. So I used read RDS to read in the saved data file for that. Something that's kind of helpful is you can do glimpse cortex, take a look at what we have here. After running everything, oh yes, so in metadata now for cortex, you can see now it's got results from the RCTD because I added it back as metadata. It has the main things to see here are that it now has first class and first type and second type. So it's going to have one of those labels from the single cell RNA-seq dataset. It's essentially saying first type is its best guess of what the majority of those cells within that the identity of the cells within the spot, and then the second type is its second best guess of like what it could also be a mixture of. So if we go back to here, yeah, we looked at cortex, and the other thing I wanted to show you was cortex data. So cortex, metadata, and first type. It'll actually show for the list essentially of all the cell identities. And there's also second type as well, but you can do summary on that to then see of the cells that we have a whole bunch. We have a lot of interneuron. We have a lot of interneurons, and then we also have some astrocytes and also oligodendrocytes as well. So first here, all I'm doing is looking at first type. Sometimes it has Na, I believe. So yeah, so some of them were not able to be. Oh no, sorry. What happens is that actually it was able to label most things, but yeah, I guess if there's any NAs, we want to make sure that they're unknown. Oh, and then what we're going to do back is to project the labels from the sketch cells to all the cortical cells. And so that gets run here. So essentially we've downsampled the data, run it on the downsampled data, and then we want to take the results and put it back to the original data set. So now we're going back to, so what we did is we were able to do that for cortex, and we're basically putting everything that was in the cortex data set and putting it back into the brain, the full data set. So a lot of the cells in brain MT filt were not run because they were not part of the cortex. Those will end up being called unknown, but everything that was run and could be labeled will get a proper assignment from the cortex data set. I think what's kind of interesting here is, yeah, so what's interesting here is that, yeah, I think we talked a bit about how there's first type and everything gets assigned its best guess. The second one that's actually guessed, that's assigned is actually, so most of these I think are neurons, but then also you actually see astrocytes showing up as a second type, which makes sense because if you're getting a tissue, a brain tissue, you're going to get a lot of neurons, perhaps in this cortex area, but then there's also going to be a lot of astrocytes and oligodendrocytes types of glia that are intermixed within it as well. So that gets assigned as well. I was able to save the data just in case. But here we're going to actually look at the different labels for the layers. And something that's interesting because we did the cortex is that there are all these interesting excitatory layer interneurons. Those labels all start with L for the different layer that that's corresponding to. Here we're telling brain empty filt to take the cells, the cell identities that are in here and label it as cells. And then we basically say, okay, we want those cells which have the names that start with letter L, and then we're going to actually plot them. So that's shown here. And then it shows the different excitatory layer neurons. There are actually 6 of them that were able to be identified and labeled. And then we can actually show here I'm just taking the first full type that we got from doing RCTD and plotting them and showing just the percentage. So a lot of the cells are as unknown because actually a lot of them were not even, are not cortex cells, not interneuron cells. But we do have a lot of neurons that were able to be labeled as interneurons, and we see a lot that are labeled as purely oligodendrocytes, parvalbumin neurons, SST neurons, etc. And then the last thing here is the showing the annotated cell identities on a spatial map. So in addition, like we've been looking at everything on top of a tissue histology, but we can actually also plot the data with ImageStimplot, provide brain empty filt data. It's going to automatically be looking at the assigned cluster or the assigned cell identity, and then you can actually plot that here. So that gets shown as just all the different interneuron layers and other types of cells that are able to be found. So I think that's everything that I've prepared today. Are there any questions? I know that we went a little bit quickly, I guess. Are there any further questions about some of the things that we covered today? Yeah, you're right. This is true. The image is upside down here. You can rotate the images with ggplot. There is actually a slight error in Seurat, which I have to report, where this actually got a little bit mirrored between image dim plot and spatial dim plot. That's a good note. Yeah, this is a package error that hasn't that other people have reported but hasn't been addressed by Seurat. It gets rotated. Um, For the unknown areas, okay, that's a good question. You can get other single-cell RNA-seq data sets, right? So for the purposes of this, we did the cortex. It's a very small area because we were interested in those like interneuron layers. But if you wanted, you could try and find another single-cell RNA-seq data set that has other cell types that you think are here labeled, and then you could run RCTD with that. So that's definitely a huge thing with analyzing spatial transcriptomics data. We're benefiting a lot um, from the single cell RNA-seq data set that's available, and that'll help you label those cells. Um, I think I kind of talked about this in part one, but one of the good things about being able to run RCTD on this is so like it's not much an issue with Visium because the good thing is we have a lot of different genes. Like we were able to observe like 5,000 different genes. So so every cell has a good like we essentially have a transcriptome for each one. But if you were to analyze things like Xenium data, where maybe you only have 150 genes, you'd really want to be doing that also. So you want to be actually figuring out what the cell identity is. And so you want to be running RCTD with that to really identify what the cells are before you make any comparisons between them. You crop the data and redid SC transform. If you don't, want to bring that content. So if I cropped the tissue data set, right, and then redid SE transform, generally, actually, I would do the cropping at the very end. So I would actually run all the analysis on everything because you don't want to have to run it on each individual component of the data set. I think I would just run it all on everything. And then if you wanted to do a analysis of that small section, then uh, then you would do the cropping towards the end. So miter filter was done really, really early because I wanted to get rid of that across the entire data set. Okay, so if single cell data for the specific species is not available, is it possible to use data sets from related species? Yeah, I think Colton might actually have done something like that because you can use a single cell RNA-seq data set. Actually, I've done it a little bit too, actually. Yeah, like we've done things where it's non-human primate. You can do it. You just have to make sure that the gene names, one, the species are similar enough that you actually think that the different cell identities and gene expression for each cell is similar enough. You'd also have to do probably some conversion between gene names, but it is technically possible. I think there is a program called Harmony that people use for that. And Colton might be able to speak more about it. Um, does Seurat work for expression data such as protein abundance or only count data? Um, I don't have any experience with protein. I I'm not sure. I'd have to do some research on that. Um, I mean, it's count data for transcripts. I guess you could maybe Seurat can deal with protein data, but I'm not really sure. Probably not. There are other packages available for RCTD. I chose this one because well, I actually don't know all the details for all of these deconvolution steps, but there are other alternatives. If you didn't want to do RCTD, you could use, there's called like cell to location. There's a bunch of other alternative methods for doing that as well. There's a slide in part one that talks about different deconvolution methods. You can look over there if you want to, if you wanted to try other methods, basically. I like RCTD because you can run it on both, like Visium data, Xenium data, etc. Visium HD analysis differ much from Visium. I mean, it's definitely way higher resolution. So I think that Well, the main thing that I've noticed with Visium HD is that it's just way, way bigger. So everything takes way, way longer. But yeah, it's very, very similar. You can do the binning in different ways. That allows you to kind of control. You're essentially getting way more, way more cells. Like you can think of these spots as almost being like it's a mixture of cells, but when you're running these analyses, it's getting treated, especially in the early pre-processing steps, as like an individual cell, so that it runs way faster. But yeah, Visium HD, it's a much more customizable, and everything will take longer. But the whole principles are all very similar in terms of all the pre-processing steps and everything. Um, I think I answered most of the questions. Feel free to email me or reach out to BCBB if you're interested. Um, And if you have a specific request for a type of analysis, I try to keep everything fairly um, general because this analysis, I wanted to just kind of give you an idea of how to pre-process the data, how to do very basic analysis like differential gene expression analysis, how to, um, you know, do all this annotation. But I didn't want to get too far in the weeds because it's very specific to your project.